Is that working?
It's working perfectly.
Perfect.
So let me
Okay.
just change here the view. Yeah, it's perfect. So you want.
Thank you very much, first of all, to the organizers, Pedro, Maria, Emmanuel, for inviting me to this nice school on the INFRACHIP project. And as said, I want to show you a little bit some of our recent work on printing for unconventional and sustainable sensors. So let's see if slides work. Okay. I think you should be all aware, or you're all aware, that we have seen in this recent decades a paradigm shift from conventional, planar, let's see if I can put a pointer, planar, bulky, rigid, invasive technologies, silicon-based mostly, to unconventional electronics. We like this work because it's electronic that has many advantages and many features. So it can be conformable, can be lightweight, deformable, and also imperceptible. And what we're also seeing recently, and also the group in Lisbon is very active in this area, is the shift to unconventional and sustainable electronics, with the idea, and we try to avoid all the electronics that creates pollution, requires a lot of energy, results in waste, plastic, electronic waste, also materials that are being mined, and has a negative impact on the environment, to electronics that has or can even have a positive impact on the environment, using natural materials that is biocompatible, maybe biodegradable, general functional, and results in something good for our planet. And of course, this allows seamless integration everywhere, from textiles to health systems, wearables, electronic robotic skins, consumer devices, and also recently this very interesting area of agri-food. So technologies for food, packaging, but not only tax, but also precision agriculture. And I think here is really where the technology can exploit its maximum benefits at the interface with the environment. And how we try to tackle this at the University of Bolzano in the group I'm heading is from the use of materials. We're engineers, right? So we're not really synthesizing materials, but rather using commercial materials or materials, let's say, developed by collaborators. So starting from the fabrication with and on sustainable materials, paper, chitosan, cellulose, to the printing, not only printing, because we work a lot also on microfabrication. Today, I want to focus more on printing, actually, to the devices, of course, sensors, biosensors, actuators, also thin film transistors and circuits, to the systems. Often hybrid systems, so requiring also, let's say, some IC, some silicon electronics, and the final application to showcase really the applicability and usability of the technology. And the approach to sustainability is double, let's say. On one side, we use electronics, which is more sustainable, but maybe not immediately completely sustainable, but let's say that has a way ahead sustainability for sustainable applications. So on the body, for fruit monitoring on plant and post-harvest, to plant health monitoring. And on the other side, I think the most powerful approach, but also the most complex approach, is to bring sustainability into the electronics, and in particular in the sensors. So use, let's say, reduce the carbon footprint of what you create, and also the waste, reuse materials to develop green electronics, and then use that to reduce the food waste. And I think that's really where the power of this technology really shows its best. And today I want to show you both approaches. So on three application use case of printed sensors for sports applications, also printed sensors for fruit monitoring using bioimpedance, for wearable plant monitoring, so on plant monitoring, and also an example from the approach of fully circular or fully green devices, and come to the conclusions afterwards. If we go at the beginning, so I think the field of sports and also clinics application is where flexible electronics has shown more examples. And here we're talking about, let's say, sensorizing a lot of different parts of the body. So from the garments to wearable watches, to salts, for measuring of a lot of different parameters that are of interest for the final applications. We, in particular, worked within this project. It's European Funds for Regional Development in South Tyrol, in a collaborative project between universities, in particular us, and also medical doctors from the University of Verona, and companies. Microgate, Microphoton Devices are two technology providing companies working also in the field of smart monitoring. Microgate provides this timing systems for all sophisticated athletes, actually, and applications. And a validation company, so company with validation of sensors for sports promoters, and also textile company. This is the project called Smart Textiles for monitoring muscle activity. And together with the doctor, when we started the project, we focused on what do we need to measure and how do we need to measure it. And we identified two approaches. So on one side, a very simple and higher TRL approach of monitoring the breathing rate, because that's an indicator of muscular exertion. I mean, the main idea was, can we monitor the muscular fatigue of athletes and provide real-time feedback before it's too late. So at first, a higher TRL approach, and the second, a lower TRL approach, more research approach, I would say, on sweat monitoring. And in particular, of sweat monitoring, we thought at the beginning of lactate, but then we identified that the real indicator in the sweat is actually ammonium ion, because that's an indicator of when we pass from aerobic to anaerobic exercise. The state of the art, the golden standard of the field are very bulky. So these bulky masks or laboratory analysis. So let's say the idea was, can we develop something that is, on the other hand, smart, textile, breathable, reliable, reusable, low power consumption, wearable, and that's how we started basically the project. The first is very simple. I mean, we are breathing, our chest is expanding and shrinking with strain that are between 4 and 10%, so pretty reasonable strains. Can we develop something completely printed? And here we choose the approach of screen printing, which is, let's say, a well-established technique in the printing industry, also for printing the logos of the t-shirts, and that was the idea. Can we develop really a t-shirt with integrated sensors and using commercially available inks? So first, screen printing a stretchable carbon ink on a thermoplastic polyurethane TPU substrate, continuing with the printing and encapsulant, and then, let's say, transferring and heat laminating the TPU substrate onto the textile and shaping it and laminating it. That's the result of the first, with this U shape that was designed in order to have the desired strain characteristic. And that's the first prototype realized, and we integrated this also into a t-shirt. We tested this clearly in laboratory conditions, looking at different textiles provided by the textile company. I tried to remove a bit of slide because it was a bit too much. But there we characterized all these different textiles. We ended up choosing one of the textiles and then testing it in this company, Promotus, together with the readout system that was developed by the company, with a protocol, again, developed by the medical doctors. That's the really strip with all the connections, but then we developed a more advanced system. What is interesting to see, again, are the resistance changing. It's a simple resistive sensor with the different phases, so resting, cycling. You see, we have this 5 minutes protocol of resting, cycling, and recovering. And from here on, look at really all the extraction of the different parameters, the breathe by breathe period, the breathing rate, of course, a coefficient of variation, and finding the patterns really to be able to discriminate between those different phases. There is clearly still work to do on that, but in parallel, we worked also on the other approach, on the sweat sensing. And we choose to work with electrolyte-gated field effect transistors, transistor-based sensing platform to be in the final use case, be integrated into a smartwatch for sweat sensing. This is how the device looks like. So we work on polyamide, 50 micrometer polyamide foil. The gate, source, and drain contacts are completely planar, so developed on the same layer. In this case, those are microfabricating. So we are talking about evaporation and lithographic lift-off. So it's not completely printed platform. The carbon nanotubes, so the semiconducting layer we choose are spray coated, so in this case, printed spray coated layers of single wall carbon nanotubes, random network, what you see here of single wall carbon nanotubes that are basically connecting the source and drain. And because the final application is testing everything in sweat, the electrolyte used is actually artificial sweat. To have this also selective for the selective sensing of ammonium ion, we use ion selective membrane for ammonium ion, an ionophore. This is how the device works very briefly. What you see here is the cross section with the channel, the carbon nanotubes, the electrolyte with all the ions, positive and negative, and the gate. So if we look at it, even if it's a planar geometry. Now, if we apply gate and drain, basically a voltage between the gate and the source, and the drain and the source, with the source grounded, particular negative voltage at the gate, because we're talking about p-type semiconductor. What is happening is that with the negative gate, we have a rearrangement of the ions. So the positive ions get attracted to the negative gate, and the negative ions on the opposite side. And so we have a variation of the conductivity in the channel, which is what we measured then in the current. Now, this leads to the creation of the so-called bi-length, which is the length basically. So we form two electric double layers, one at the interface between the gate and the electrolyte, and the other at the interface between the semiconductor and the electrolyte. And what happens basically is that the electrolyte, at least the bulk of the electrolyte, behaves like a gate dielectric. It's clearly not a gate dielectric. But there is basically no conduction anymore in the bulk of the dielectric. And you have effects which are interesting here at the two interfaces at the bi-length. Now, if you use an ion selective membrane, what happens is that you allow only the passage of the ions of interest, in this case the ammonium ion, through the membranes, and only those can interact with the semiconductor. And interact and thereby change the conductivity and have a variation in the currents that can be detected. And that leads to the sensor characteristic, basically. This is quickly how the devices are made. As I said, the electrodes are microfabricated. We have a spray deposition of the carbon nanotubes, the water-based carbon nanotube ink that we use with commercially available single-wall carbon nanotubes. We drop cast the membrane that is synthesized. There is a membrane conditioning step that is always required for the devices. You see here the devices with the big gate pads. It's not really used, but it's a geometry that we use when we're functionalizing also the gate. So in this case, there is no need. But this is the gate. Those are the two interdigitated electrodes for the source and drain. And then we have the electrolyte contained in a chamber that we create here. As you see, in the carbon nanotubes are covering basically interconnecting the source and the drain. This is how it looks like. That's the membrane. So before the electrolyte. And what you see here is actually the small chamber that is containing the electrolyte, and everything tested first clearly with a probe station and parameter analyzer. We have to do a baseline fitting of the devices because the carbon nanotubes have some variation, especially when we have the establishment of the two electric double layers. But what you see is once we do we account for roughly an hour of stabilization phase. What we can then remove is that baseline. And then what you see here is the variation of the current upon the variation of the concentration of the ammonium ion, which is pipetted inside the electrolyte concentration. Being an electrolyte-gated transistor, we work at very low voltages. And so that's one of the advantages also. So it's a very low power technology. This is actually the interesting process window of the devices, what we're interested for these applications. And you see also the calibration curves of the devices, first in DI water, but also in artificial sweat, especially when we have conditioning of the devices, which is very important, the membrane conditioning. So pretty linear behavior with a good sensitivity and also good linearity. So with this, we continued, actually, the work of Mattia continued at APF Fellows in the group of Professor Sandro Carrara in trying to make a full system, right? Because that's the device is only, the sensor is only a part of the final application. I think the school is also an integration, which is a challenge, actually. And in particular, he worked also on the electronics, on the readout electronics. You see is a hybrid system, so with components in this case, but on a flexible PCB. So this has the transimpedance amplifier clearly needed for a current source like our device, the analog to digital converter, the Bluetooth connectivity, and the battery. And that's what you see here. Of course, you need also the microfluidic, eventually iontophoresis. So there are more integration challenges, of course, required. Yeah, you see also this is clearly working also when bent in a final application. So we did also some static bending tests of the final devices. Good. Let me switch gears. I am looking also at the time. Beside wearables, we have been focusing a lot on agri-food and in particular on fruit monitoring using bioimpedance. Bioimpedance is a very old concept or an old technique, but it's gaining a lot of momentum. I mean, it has been used and it's still used a lot in on-body applications, but it's very interesting for agri-food applications. In general, for biological tissues, so for fruit quality evaluation, also plant health monitoring, but again, it's in many more. So we are focusing mostly on this application, fruits and plants, but today I want to focus more on fruits and show you a little bit of the work done. The concept is very easy because, I mean, you're basically applying an AC voltage to a biological tissue and measuring the current that is coming out. And extracting basically the impedance, so the complex resistance, so the magnitude and the phase of the impedance. And as engineers, try to develop circuits, equivalent circuit models that describe you the biological tissue and try to fit these circuit models to the physiological characteristics of the biological tissue. So make a biophysical model, basically. The model that mostly describes these types of tissues is basically the Cole model, which accounts for an extracellular fluid resistance, an intracellular fluid resistance, and a capacitance of the membrane. So resistor in series with the parallel of resistor and capacitors. And the interesting bit is that because you sweep the whole frequency spectrum, maybe this I forgot to say, but you're sweeping from low to high frequencies, what happens is that you have different behaviors. So on one side, at low frequencies, the path is in the extracellular. So that happens. It's not able to pass through the cell, but it goes around, right? Whereas at high frequencies, the current is able to pass through the membrane. And so you can explore with the different frequencies, different behaviors, basically. And of course, all the biological tissues have a water content, and therefore they have a conductivity. How do we do that? We take the tissue, or let's say the biological sample. In our case, in this case, is an apple. We use mostly conventional ECG electrodes, so silver chloride, portable impedance analyzers, and then of course we have our graphical user interface. But what I want to show you before is the power of the technique, but talking about printed electronics, I want to show you how we can also print dry electrode smart gloves for the application. So let's have a little bit of what the application can do, but let's focus then on the printing. We have been exploring this for a lot of different things, both post-harvest, on-plant, and in this case, in this project, Sundus has been exploring fruit damage detection, internal browning, with the technique that is, again, non-destructive, because you apply the electrodes at the outer part, you measure, and you don't need to cut the sample to see if there is internal browning, which is a big issue in apples, at least. And what happens is, of course, if the sample is healthy, and again, how this was done in the first test is labeling. So saying those samples, cutting also, those samples are healthy, those samples have internal browning. And of course, you have different behaviors based on this, because with damaged, you have a cell membrane which is disrupted, you have intracellular fluid that is moving outside the cell, and so on. What you see here is magnitude and phase for healthy, slightly brown, and severe. Again, we did labeling here, but now that we know how this behaves, we can also use it without need of cutting and destructive methods. It's difficult to see because there is clearly variation that you typically have with biological tissues, but the interesting part is that now if we focus on some specific frequencies, in this case, frequency, I think that's 30 hertz, you can see again that healthy have a higher magnitude, whereas internal browned apple have a lower impedance. Now we went on with the modeling of the equivalent circuits and finding, statistical differences. There are statistical differences. I removed the slide, but I have it in the backup in case you're interested. So I want to show you again also maturity and then the printing. So for what concerns maturity, this is clearly an important topic, both on plant, post-harvest, depending on the type of fruits, if they mature completely on plant or they mature also later. And here again, doing several studies, you can see that there is a clear pattern and differences in both the magnitude and the phase of the bioimpedance with various samples that have been measured, statistically relevant samples. Now, the technique is powerful. We published several papers on the use and also the use of machine learning in order to look at classification of this data, because it's a lot of data. Lots of samples over time, different classes of data, lots of frequencies. So it's very nice then to apply machine learning and work on the data. And that's what Pietro is doing in the group. But now I want to show you how we can make this more applied, how we can really bring it to the final application using printing technologies. That is the topic of today. Concept is, can we make some smart gloves that are able to incorporate bioimpedance electrodes and be connected to a portable analyzer and be used then throughout the whole chain, so from the field to the processing. So say supporting on-field harvest, but also post-harvest quality sorting, which is very important. And how we did this is to realize a smart glove with carbon nanfiber TPU electrodes printed and then connected to a portable impedance analyzer. That's a silicon system. And then in the future have also this fruit quality prediction and classification model via machine learning. So that's how we did it together with the Istituto Italiano di Tecnologia, the group of Nassi Attanasio, with spray coats of a particular ink formulation that was developed there, of carbon nanoflakes, TPU and natural rubber, and a particular shape for the electrodes. Besides, let's say, the SEM images that you see here, this is how the sample look like, with, of course, this connection. And I think here it's where we need a lot of work in the field on the connection and the integration. We did a lot of electromechanical characterization, both static and dynamic. Of course, if it's a glove, it will have a lot of stretching application. You see here there is also the dynamic stretching up to the breaking point, but with good performance up to 2000 stretching cycles, and also full recovery. You don't see it from this picture here, but there is also full recovery after stretching with our custom-made bending, twisting, stretching machine. We did all the electrical characterization. So what you see here is magnitude and phase of the ECG reference electrodes. Of course, that's the golden standard in bioimpedance. The fabric with and without gel. So that's a big topic, the use of gel. Again, we would like in the end to have something without gel, completely edible, ideally. So this is also, I think Mario also showed recently some nice edible electrodes, myocaroni, for bioimpedance. So that's a big topic. If you want to have good measurements, you still need to rely on gels. And that's a big issue. So what we show is that our fabric with the gel behaves pretty good up to 500 kilohertz. But that's fine, because actually with our portable impedance analyzer, the limit actually is 100 kilohertz. So we can use well our electrodes, fabric electrodes, but with the gel. And we did also measurements on plants for this, showing basically that we can measure. You see this integrated with the portable analyzer. We can measure on plant the impedance of strawberries, which are very tiny fruits actually, and with having the curve with the expected trend. So Pietro showed this recently in JFlex and the conference FETC in Bologna. I'm looking at the time. I'm trying to go quicker, but Pedro interrupt me. We started a little later, right? So
no, you still have time. We started a little bit later. Don't worry. Take your time.
Good. I want to show you also a little bit of the work on plant monitoring, in particular using very simple sensors. So those are impedimetrics, so again, impedance, printed sensors on cellulose, on a filtered cellulose paper using P.PSS interdigitated contacts that are completely printed in this way with a different printing technique. We're talking about dispense printing. That is a digital printing technique, and we're looking at the variation of the impedance with the relative humidity. So the concept and the sensor extremely easy. But the idea here is to go really on the field, right? Because testing in the lab is easy. I mean, you have impedimetric change with the relative humidity. You characterize your sensor. You make your calibration curves using a climate chamber. But then going in the field is something complex. So what Saira showed is that we can test this on plants, in this case, tomato plants, in various locations, taking benefit of the fact that the sensor is really flexible. It can be attached on the surface, on the leaf, on the bottom of the surface. So you can compare it with the reference sensors that are in the plant environment, but you have an additional information by having the sensor directly attached to the interface of the plant. So she went from, let's say, the climate chamber to the office, tomato plants in the office, to the greenhouse, using a larger sample of tomato. We have access to this greenhouse close to Bolzano, and looking that it can track very well reference sensor, but the information on the leaf, there is more information on the leaf. So that's where she's now looking into, can we really trace a potential transpiration on the leaf with these sensors? So with these good experiments in the greenhouse, she went to the open field. So we have access through our partners, Limburg, to this, it's called Lido, Limburg, which is the place where it is, Limburg Integrated Digital Orchard. And it's an intelligent orchard, basically, with apples, you see, but not only. There is a LoRaWAN connectivity, there is electrification, so it's an open field where you can test sensors. And she has been running tests over the summer, actually. I think they're still there, with, of course, a system, the readout electronics that is, and the communication part that is clearly standard, conventional, and monitoring basically both reference and printed sensors. So still work ongoing, but we showed that it's possible to use these sensors in the open field, and now there is a lot more to do on the encapsulation, on various things, because in the field you have clearly everything that is happening. Yeah, so that was all here. I'll briefly show the last approach with biodegradable thin film sensors, in this case microfabricated. This is part of this national print project called Bytes for IoT, together with the University of Modena, Reggio Emilia, and Venice. The group of Giovanni Salvatore. And here I'm trying to go back to my old, let's say, love of the PhD in thin film transistors. We aim to develop fully biodegradable thin film transistors on chitosan. That is, let's say, a substrate derived from the crab, and it's completely biodegradable. And here we're working on the materials. We started simple with sensors, developing simple resistive temperature detectors, but also thermistors with molybdenum as the metal layer and indium gallium zinc oxide as the semiconductor, with the idea now also with the dielectric we are optimizing to develop fully biodegradable transistors and circuits. In parallel, in Modena, they're working on the modeling of the devices. This is dissolvable or transient, so basically it can dissolve in lime solution, deer water, and also vinegar. Very recent results that Ahmed is now wrapping up for a publication. And this brings me to the conclusion. I think I'm on time. Of course, I have to thank a lot of members of the group, especially Martina, Mattia, Bulia, Sundus, Pietro, Saira, Ahmed, Enrico. Not only a lot of collaborators that I showed. You see the main collaborators here. And this I didn't show because it was too much time, but we're working also on bioimpedance electrodes from onion skin. So this is also something I decided to remove last minute, but I have some backup. The group is still very big. We just moved into the new Faculty of Engineering, and all the collaborators, many collaborators, and the funding agency. And you for the attention.
Thank you, Luisa. Amazing talk. Thank you so much. Really, really interesting topics brought into this school. So I would open the stage now for questions. So if you have any questions for Luisa, please use the raise your hand feature in
I see some questions in the chat also.
Okay, I'm searching for that as well.
I can reply there. So why do you use carbon nanotubes instead of graphene? But that's a good question. The use of carbon nanotubes is historical in our group because so the group started also together with Paolo Lugli, who was formerly in Munich, working a lot on carbon nanotubes. And when we started the labs from scratch, we had to start from something we knew how to work on. So more than any other, let's say, more scientific answer, there is clearly this historical reason. Also is a nice material because it's sensitive to many things, maybe too many, because besides being printable, it has a temperature dependence, it has a gas sensing characteristic, so it's definitely a nice materials for this. I historically come from metal oxide, so I'm not a carbon nanotube person. Yeah, I hope I replied to this one. then can PVD techniques be used to create flexible and stretchable electrodes for bioimpedance-based smart scrub instead of printed conductive inks? I mean, PVD techniques are very powerful, especially sputtering, I would say. We are using a lot of sputtering of materials, also biodegradable materials like molybdenum, magnesium, and so on. Also on let's say weird substrate and textile. My colleague Nico Münsterida here is exploiting a lot of sputtering on threads and textile, so it's definitely a powerful technique and can be used instead of printing. Of course, it's always a trade-off, right? I mean, printing is lower cost, lower infrastructure required. Also, the inks are. I mean, it's always a debate, right, between the two words. I'm personally a little bit in between, so I use what makes more sense for the specific application. Then I see another question, and I think, Emmanuel, you have two questions, right?
Emmanuel has two questions.
Yeah,
go ahead.
So one is about the sensor that you showed in the plant. So it's really important, the permeability of the membrane, right? Because you need to take into account the external. So imagine light and so on, and if the plant also don't absorb the right energy, so sunlight and other even humidity and so on, it can affect the measurement of the sensor.
Yeah,
right?
So it's very difficult to go on field exactly because there are many parameters to take into account. So Saira is testing now encapsulated sensors with PDMS and other materials. I mean, in this case, we wanted to exploit the fact that water is absorbed by the substrate, right? So it's part of the device characteristic itself. But then it's clearly questionable in case of water and other situations which occur in the field. So there is lots to do. But here we went really straight. Let's try to test a sensor that works in the field. We have another project again together with Limburg, a PhD project. It's a student who comes with a background in horticulture. So he digged into engineering, but coming with a clear understanding of the physiology. And he is looking into stretchable sensors for on-plant growth monitoring. So in an orchard with apples, he's creating kind of a socket for measuring from the very beginning to the end. It's 100% strain, so much more than what I showed at the beginning, so way more challenges. And then transpiration of the apple during the growth, light that needs to come through. So all of these parameters from the beginning. So he's doing some prototypes.
Of Mario too, right? Mario Caironi, he also have something related to that.
He showed the edible bioimpedance electrodes, right? Recently.
Yeah.
He's doing lots of work in that.
And one way to growing of the apple, too.
So now Wim, the firm, has a similar project. That's why also Michele is there. So in Belgium, right? And so he started to look at all these requirements from the application side, being also having a master in horticulture, right? And then trying to sort the engineering around the requirements from the application. That's not easy.
Yes.
Yeah.
And the second question was about the biodegradability. So sure, it can be biodegradable in water and other solvents. But the point is, if you want to have the sensor imagine on a crop, it can take, imagine, like around 3 months since you plant until you get something out. So the term of biodegradability could be like if you can degrade over time. So after 3 months, it starts to degrade a lot. I think that is also one point to take in consideration. It's good that we can degrade immediately, but if you think in a final end, it should be biodegradable, but in a short period of time, let's say.
A good point. I mean, here we are not looking into time scales. We're looking into, can we make this fully biodegradable transistors with oxides? So it's not about, the time scale. Clearly, that depends on the application. I don't think we need always biodegradable stuff. I think it's fancy for the papers to publish, but not necessarily we need something biodegradable, right? So, and also there is a lot of confusion on terms. What is biodegradable? What is transient?
Exactly.
What is all the other words that we see. Here we are doing it a little bit in an ignorant way, I would say. So can we make them degradable? We're exploring different materials, basically. So it's more an explorative study on our capabilities also to make these devices.
Yeah. I think that is the first point that you made. That is what we should be made.
Thank you. So we have an extra question here from Julio in the chat about advantages of using FATs.
So what advantages have to use FETs for for ammonium ion instead of solid state ion selective electrodes? We use also electrochemical electrodes, so both 3 electrodes, potentiometric. The advantage of having a transistor-based device is that it's a transistor, so you have an inherent amplification. I keep having these questions also from companies, what to use. Transistors are better, I would say, because you have an inherent amplification, but they have a lower TRL still. I mean, carbon nanotubes for sure. There are many technologies around. I mean, Luisa Torsi and the group have been working on organic P3HT, so I think that has a higher TRL. In terms of electrolyte gated field effect transistors, now there are also OECTs. Transistor is definitely better, especially if you want to sense smaller concentration in very noisy environment, complex matrices. But let's say potentiometric sensors or electrochemical sensors are more established, have a higher TRL. So when we look into industrial project, I never propose transistor-based platforms, honestly. I think there is still a lot of way to go until this becomes really mature enough. Yeah.
Amazing. Luisa, thank you so much. It was amazing to have you here. Thank you so much for participating.